Hi students, welcome to Aakash PG Plus. I am Dr. Gurman Gill, faculty for Biochem. In this, we will be quickly revising various facts and concepts to solve any type of question asked in your exam. We will also discuss various types of PYQs and how to answer them. And I strongly recommend after watching this, you should attempt the practice quiz given in app so that you can see where you are lagging and plan accordingly. So let's begin. In this, we will be discussing about three very important topics, that is insulin, alcohol metabolism and heme. So first one is insulin. As we know, many questions asked about insulin and some are really tricky also. So these types of questions are frequently asked when there is low insulin glucagon ratio. Which of the following enzymes is stimulated? So here insulin glucagon ratio. In some questions it is asked about insulin. Some questions it is asked about glucagon. Sometimes it asks which enzymes are active in fasting state, which enzymes are active in fed state. So to answer this, first of all, you should understand that whenever we take food, insulin is secreted. And insulin mainly acts by dephosphorylation of enzyme. So how will it dephosphorylate enzymes? By decreasing the level of cyclic AMP by activating phosphodiesterase. On the other hand, when we are fasting, at that time our body secretes glucagon and it mainly acts by phosphorylation of enzyme. How? By activating cyclic AMP. Now you can see they act opposite to each other. One is acting by dephosphorylation and other is acting by phosphorylation. Further, in our body there are certain enzymes which are active in dephosphorylated state. So what will happen? They will be active whenever we take food or whenever there is secretion of insulin, and they will be inactive whenever we are fasting or glucagon is secreted. So because these enzymes are active in dephosphorylated state. Similarly, there are certain other enzymes which are active in phosphorylated state. So when will be they active? They will be active in conditions like fasting and glucagon and will be inactive in case of food and insulin. So you can see both of them, insulin and glucagon, fasting and fed state, act opposite to each other, whereas food and insulin goes together. Similarly, fasting and glucagon goes together. Now further, what types of enzymes or what type of various body processes are active after eating food, that is when insulin is more. So whenever there is more insulin, that is after taking food, insulin glucagon ratio is more, first of all glycolysis will be activated. So glycolysis helps to utilize the glucose. So the enzymes or main enzymes of glycolysis, that is phosphofructokinase, glucokinase and pyruvate kinase, are activated by insulin. So here you must note that it is glucokinase, not hexokinase, and glucokinase is present in the liver. So it will be active when we have taken food and there is sufficient glucose in blood. So liver glucokinase will be activated in presence of food only because it is dependent upon insulin. Similarly, the pyruvate dehydrogenase complex will be active, which further convert pyruvate to acetyl-CoA, and HMP shunt will be active. So first of all, after taking food, insulin is secreted and our body starts utilizing glucose by glycolysis and HMP shunt. Further, if there are still energy rich condition, our body will start forming larger molecules like glycogen, fatty acid, cholesterol, triacylglycerol and protein. So here you can see, in presence of food, that means we are rich in energy, our body will synthesize bigger molecules. So what are the enzymes activated? Now you know that for glycogen synthesis, the main enzyme is glycogen synthase. So it will be activated in presence of food, that is in presence of insulin. So insulin will activate glycogen synthase. Similarly, the fatty acid synthesis enzyme acetyl-CoA carboxylase will be active. Similarly, for cholesterol synthesis, HMG-CoA reductase will be active. So here you should know it is reductase, not synthase. HMG-CoA synthase is for ketone bodies. So reductase is active in presence of food and insulin because our body tend to synthesize bigger molecules when there is sufficient supply of glucose. Then triacylglycerol synthesis will be activated by activating lipoprotein lipase, and lastly protein synthesis will be activated by increasing the translation process of the proteins by insulin. So these are the enzymes which are active whenever we take food or whenever there is secretion of insulin. So you should remember, if asked which enzymes are after taking food, or which enzymes are activated by insulin, or which enzymes are inactivated by glucagon or fasting state. So all these enzymes will be inactivated or in the presence of glucagon or fasting state, because glucagon and fasting acts opposite to food and insulin. So whenever we have taken food, our body will activate glycolysis, HMP shunt, and will try to synthesize larger molecules like glycogen synthesis, lipid synthesis and protein synthesis. So these are the enzymes which are activated. Then opposite will happen when there is fasting state. Now in fasting state we are poor in glucose, that is insulin levels are low and glucagon levels are high. So what will happen? First of all, our body try to synthesize new glucose, that is by process of gluconeogenesis. So the enzymes, main enzymes of gluconeogenesis will be activated. Those are pyruvate carboxylase, phosphophenol pyruvate carboxykinase and glucose 6-phosphatase. So, and then it will start breaking down the larger molecules which were built in presence of food and insulin. Like glycogen synthesis was activated by insulin. Now there will be glycogen breakdown in the case of fasting or glucagon. Similarly, lipids will be broken down by activating hormone sensitive lipase, and our body try to synthesize new sources of energy like ketone bodies by activating HMG CoA synthase. So students, you should know that here it is HMG CoA synthase, which is also known as HMG CoA lyase, which is activated in fasting, whereas in case of food, which one is activated? HMG CoA reductase, which is key enzyme for cholesterol synthesis. So in the case of fasting, there will be, our body will try to synthesize new glucose as well as break down the larger molecules which were built in presence of food or insulin. So these processes will be active in fasting or in presence of glucagon. And if asked which enzymes are inactivated in presence of insulin or food, so these will be inactive in insulin or food because they are activated only in the presence of fasting or glucagon. So in this way, if you understand which process is being activated, that is which is synthesizing larger molecules, that will be activating in food and insulin, and those which are breaking down, that will be activating in fasting or glucagon to provide the energy required by our body. So next is, you should know at what time of after how many hours of fasting which process is activated. Now we know that in case of fasting, our body first will break down glycogen, proteins, lipids, and also will try to synthesize new glucose. But what is the sequence? So this is important because this can be asked, at what time what is activated. So students, till 4 hours after eating food, we are rich in glucose. But after 4 to 18 hours, when glucose levels starts lowering, at that time, first of all, glycogen is broken down. So you should remember, it's not gluconeogenesis is first, it is glycogen is broken down by activating glycogen phosphorylase. And after 18 hours, when glycogen stores are depleted, so what will glycogen do after breaking? It will provide glucose to our body. Why? Because we are fasting and we don't have sufficient amount of glucose. So this glucose will be provided by breakdown of glycogen, which is known as glycogenolysis. And after 18 hours, 2 days, after 18 hours, what will happen? Glycogen stores will be depleted. At that time, our body will try to synthesize new glucose. Why? Because RBCs, our brain, are dependent on glucose. They need continuous supply of glucose. So when glycogen is depleted, it cannot provide further glucose. At that time, our body will activate gluconeogenesis, that it will try to synthesize new glucose. How? By activating the enzyme of gluconeogenesis. So if asked which enzymes will be activated after 18 hours or before 48 hours, so these are the enzymes of gluconeogenesis, whereas 4 to 18 hours, the enzymes activated of glycogenolysis, that is glycogen phosphorylase. After 2 to 5 days, after 2 days of fasting, then our body will try to switch on other sources of energy. So it will start breaking down lipids, that is fatty acid oxidation will begin, and lipids will be broken down by activating hormone sensitive lipase, and then it will, from the breakdown of product of lipids, that is acetyl-CoA. As acetyl-CoA cannot be entered TCA cycle, it is diverted towards ketone body synthesis. So, because of fasting, TCA cycle also has limited supply of oxaloacetate. Further, oxaloacetate is used up for gluconeogenesis. So, this acetyl-CoA is diverted towards ketone body synthesis, which are also a source of energy. So, after 2 to 5 days, main source of energy are lipid breakdown and ketogenesis. And after 5 days, protein breakdown will begin and death will occur when tissue proteins are catabolized, not replaced, because they are required to survive. So, protein breakdown will begin after 5 days. So, this you should remember. So, first 4 to 18 hours, first there is glycogenolysis, then 18 hours to 2 days, or you can say 48 hours, there is gluconeogenesis. 2 days to 5 days, there is lipid breakdown and ketogenesis, and after 5 days, protein breakdown will begin. So, this you should remember because many questions are asked at what time which process will be activated. So, further, this is also important, you should know which organ or which cell need which type of energy. So, when we are well fed, that is 1 to 4 hours after taking food, almost all organs are dependent upon glucose because glucose supply is maximum, except heart, which prefers fatty acid. Heart prefers fatty acid over glucose, and all other uses glucose for energy. But what happens during early fasting? So, at that time, RBCs are dependent on glucose as well as brain is dependent on glucose. So, these two all require glucose, whereas all other shift to fatty acids. So, they all other shift to fatty acids. They start using fatty acid for their energy. And in case of starvation, what will happen? RBCs always need glucose. You should remember why they always need glucose, because they have only cytoplasm. And in cytoplasm they can only use glucose by the process of glycolysis. So, RBC, whether we are fasting, whether we are starving, the RBCs need glucose to survive. So, glucose has to be supplied continuously to RBC for the survival. And the brain first uses glucose, but later on it shifts to ketone bodies. But here you should remember, brain never use fatty acid. So, you can remember there is not no fat brain, or there is. So, brain do not like fatty acid. It either uses glucose, or then during starvation it will shift to ketone bodies. It will not use fatty acid. So, this you should remember because this can be asked. Then the heart uses fatty acid, but even during the starvation it will also shift to ketone bodies. So, ketone bodies are mainly used by brain and heart, which are two important organs and very vital organs. The liver uses amino acids, and skeletal muscles also uses amino acids or ketone bodies according to the availability. So, you can see the RBCs always use glucose, whereas brain shift to ketone bodies but not use fatty acid. So, brain never uses fatty acid, it either uses glucose or it uses ketone bodies. Heart preferred source is fatty acids, but it can shift to ketone bodies. Liver uses glucose, but when there is fasting it shifts to fatty acid, then it can shift to amino acid also, and skeletal muscles also for fatty acid, and then amino acid or ketone bodies, whatever there is availability. So, this you should remember which organ or which cell require what type of source during fed state, fasting state and starving state, as it is important. Now, let's discuss various types of questions which are asked. So, these are one-liners. So, enzymes active in fed state. So, we have remembered that whenever we take food, insulin is activated, and insulin mostly activates enzymes by dephosphorylation by decreasing the level of cyclic AMP. So, these are the enzymes of glycolysis. So, these are the enzyme of glycolysis, HMP shunt. Then there is synthesis of bigger molecules like glycogen. So, glycogen synthase will be activated. Cholesterol by activating HMG-CoA reductase. Triacylglycerols and even proteins will be synthesized more by increasing their translation process. Then which enzymes are inactive in the fasting state or during glucagon or phosphorylation? So, questions are asked like this. Which of following will be active in fasting state? Which of following will be active in presence of insulin or high insulin glucagon ratio? So if you understand which process will be active in presence of food and which process will be activated in fasting, you can easily answer them. So the enzymes of fasting, we need gluconeogenesis. Because why? Because RBCs and brain require continuous supply of glucose. Then we need the breakdown of glycogen, that is glycogenolysis. Enzymes of glycogenolysis are activated. Then lipolysis is activated. So in this way you can remember. Then ketone body synthesis. So the enzyme is HMG-CoA lyase or synthase. So this you should remember that for ketone body synthesis, HMG-CoA synthase is activated in fasting condition, whereas HMG-CoA reductase, which is for cholesterol synthesis, will be active in the whenever we take food or in presence of insulin. Now the question is source of energy after 18 hours of fasting. So we know the first source of energy in case of fasting is glycogen, that is glycogen breakdown. But after 18 hours glycogen breakdown is glycogen stores are depleted, so our body is dependent upon gluconeogenesis. So questions like this you should know. Then liver cannot use. So which type of source of energy liver cannot use? We know that liver cannot use ketone bodies because it lacks which enzyme? Thiophorase. So liver cannot use ketone bodies. Then brain cannot use. We know brain is not fat, so it cannot use fatty acids. RBCs can use only, we know they only use glucose because they have only cytoplasm. Then the question is after 48 hours of fasting, source of energy is. Now we have discussed that in first 18 hours it's glycogen, then 18 hours to 2 days, that is 48 hours, it is gluconeogenesis, and after 2 days to 5 days it is mainly lipolysis, that is lipid breakdown, as well as ketone body synthesis. Then question is which of the pathways are active with high insulin glucagon ratio? So this we have already discussed. These are cycles of glycolysis, HMP shunt, glycogen synthesis, cholesterol synthesis, and even protein synthesis. Then in fasting level, cyclic AMP level is. Now what will happen to cyclic AMP? So cyclic AMP basically phosphorylates the enzyme. So in fasting, when we know when there is glucagon, it activates by phosphorylation. So cyclic AMP will be decreased or increased? It will be increased, and only it can phosphorylate the enzyme and activate it. So in fasting, cyclic AMP levels are increased, whereas in case of fed state, its level are decreased. So this was various questions which are asked on insulin and glucagon and very frequently asked. Then there are questions which is very easy and very frequently asked. All of the following uptake glucose independent of insulin except. So we know which organs are dependent for glucose, for insulin, for glucose uptake. Those are skeletal muscles, cardiac muscles, and adipose tissue. So this is important because this is very important, and it was also recently asked in your NEET exam. So you should remember insulin dependent organs are skeletal muscle, cardiac muscle, and adipose tissue. So they are dependent on insulin for glucose uptake. So the process you should also remember that this is the normal beta cells of pancreas. So are beta cells of pancreas dependent on insulin for glucose uptake? No, they are only skeletal muscle, cardiac muscle, and adipose tissue. So they will quickly take the glucose whenever there is, and glucose will undergo glycolysis, and what will happen? ATP levels will increase. So if ATP levels will increase, what will happen? ATP potassium channel will be closed when there is sufficient level of ATP. But to maintain the electronegativity inside the cell, when potassium channels are closed, calcium channels open. So when calcium channels open, there will be increased level of calcium in the cell, and this calcium cause exocytosis of insulin from the pancreas. So question can be asked about this. What is the process of insulin release? So it is due to increased level of calcium which cause exocytosis of insulin. So this insulin then will act on the cells which are dependent on insulin for glucose uptake. These cells are dependent on glucose because they need GLUT4 transporters for uptake of glucose. So normally GLUT4 transporter is present inside the cell, but when insulin binds to its receptor on the cell, this GLUT4 receptor is transported to the surface of cell, and when it fuses with the surface of cell, it can intake glucose inside the cell. So in this way, glucose is supplied to the cells which are dependent on insulin for glucose, that is by GLUT4 receptors, that is skeletal, cardiac as well as adipose tissue. So normally their receptors which are responsible for intake of glucose are not fused with the cell surface, but in presence of insulin, when it binds to the receptor, these GLUT4 receptors will bind to the fuse with the cell membrane and will take the insulin inside. So this is you can see this is a normal process. When insulin binds to its receptor, GLUT4 fuses with the cell membrane and takes the glucose inside. But in type 1 diabetes, when insulin is not formed, so insulin cannot bind to receptor and GLUT4 cannot fuse to the cell membrane, so it cannot take glucose. Whereas in type 2 diabetes, there is resistance to the insulin to its receptor. So insulin is present, but it cannot bind to its receptor. So again, when it cannot bind to receptor, GLUT4 receptors cannot fuse and glucose cannot be taken in. So now the question asked is, in beta cells of pancreas, exocytosis of insulin is stimulated by influx of, it is calcium. Why? Because due to glycolysis, when ATP levels increases, potassium channels are closed. So to maintain the electronegativity, the calcium ions enter and it will cause exocytosis of insulin. So next the question asked is, insulin dependent glucose uptake is by, so this we know they are GLUT4 transporters. So this you should remember, and it is very easy, asked many times. Then insulin dependent GLUT4 transporters are present in, we know these are skeletal muscles, cardiac muscles as well as adipose tissue. After, now the question can be asked like this also, after overnight fasting, level of glucose transporters are reduced in. So here you should know that if insulin is not there, if person is fasting, what will happen? Insulin will not be secreted. So only when there is food, there is glucose, pancreas cell then only will release the insulin. So if there is no food, there is fasting, insulin will not be released. So what will happen? The glucose transporters will be reduced in these cells also, skeletal muscle, cardiac muscle and adipose tissue, because they need insulin so that their GLUT receptors can fuse with the cell membrane. Now further question is, glucose transporter in myocyte is stimulated. Again it's by insulin. Myocyte is used for muscles. So yes students, you should understand that whenever there is sufficient supply of glucose, then only these muscles and adipose tissue take up the glucose. Otherwise they do not use glucose. They spare this glucose for important organs like brain, RBCs, which require continuous supply of glucose. So these muscles and adipose tissues only can use glucose when there is sufficient supply, when the fed state. But in fasting state they will switch to other source of energy, because they spare this glucose for RBCs and brain which require it. And you should know, and I strongly recommend now to attempt the practice quiz given in the app, so that you can know where you are lacking and work on that area. That's all the best. Thank you.